assume
This
everybody's
conference will now be recorded.
where things get a little more complicated is in this second part. Well, excitation, contraction, coupling part is really not equivalent to anything that we've talked about before. So what we're dealing with here, we've got a series of individual muscle cells, or these all marked out by a pound, right? And at each of these muscle fibers, we have an individual synapse, an individual axon terminal, a synapse with that muscle cell, so on and so forth. We call this, sometimes we call this a motor end plate, is another descriptor for the synapse itself. Okay, but this is where we transition really from the first part of this, right, the phase one, and move into phase two. So at phase two, at the start of phase two, right, we bind to acetylcholine, we open sodium channels, right, we generate an action potential, essentially the same way we'd see it anywhere else, right? And that becomes very important for us because we're going to use that electrical energy then to accomplish the steps that lead towards the coalescence, maybe there's a word, maybe a bad word, the coming together of each of the Z discs in every one of our sarcomeres. This whole process is going to be, this part of the process anyway, largely familiar to you, again, because it's just generation of action potential. But we've got three stages to that. End plate potential, depolarization, and then repolarization. Again, if you remember how neurons do this, this won't be substantially different for you. So if we begin at the beginning, I guess best way to put it, right? Acetylcholine binding, right? The neurotransmitter crosses the synaptic cleft, bind to the sarcolemma, okay? Opening ion channels just as an action potential does in a neuron, okay? Your sodium channels open, sodium floods in, positive charges flood in, that electrical energy then moves on and generates the action potential farther down, right? The whole depolarization part of this. And just like a action potential propagates down a axon in a neuron, the action potential propagates along the sarcolemma and a muscle fiber. Again, by the exact same set of processes. And there's nothing any different here whatsoever, right? We still have threshold, it's still at about somewhere around -60, -55, right? It's still in that neighborhood, okay? It's still required to cross threshold to generate the action potential next door, right? All of those things, the fact that it's unidirectional, the fact that it is unstoppable, all of these things, again, exactly the same. Now, where things are going to become different for us, and it's going to kind of move forward a little bit, is when we actually arrive at the T tubules, right? Remember those canals of the sarcolemma that pass across the cell membrane? The action potential will travel down those two, and that's where things become different, okay? And again, just like in a neuron, right? The phase following depolarization is repolarization. We have a refractory period where we're non-responsive. All of these things, exactly the same. I feel like I'm repeating that a lot, but it's true, right? And so if you remember those things from before, from like 2 months ago, good to go on this one, right? So to see it in action, right, we have a nerve impulse, action potential traveling down our axon faster than that in theory anyway, right? We arrive at the motor end plate, okay, we release the acetylcholine. Acetylcholine binds to our muscle fiber through the sarcolemma itself, opening sodium channels, generating an action potential. Everything's good. Okay. It's at this point that we need to start digging into and looking at some of the stuff inside of the cell. Specifically here, P-tubule, cytoplasmic reticulum, and the combination of those two things that we call the triad. Okay? So if we take a slightly closer look here at a triad, okay, we're going to find is that they're actually directly connected to one another. Okay? There are these voltage-gated channels that connect the sarcoplasmic reticulum to the T-tubule. Okay? Behind these voltage-gated channels, we have calcium being actively pumped into the sarcoplasmic reticulum and stored there. Okay? And stored right by these voltage-gated channels, right, and held right outside of them by a protein called calsequestrin. Okay? So at rest, right, we are preparing ourselves for the release of this calcium. Okay? We're pulling it out of the sarcoplasm, hiding it in the sarcoplasmic reticulum, and removing it right next door here, tying it up in this calsequestrin, right next door to these channels, so that when they open, we're off to the races. Again, here at rest, holding it back, storing it, getting it ready. Now, as the action potential travels down the sarcolemma, it propagates from one end to the other. It's eventually going to encounter a T-tubule. Okay? And as it travels down the T-tubule, it opens these voltage-gated channels. This is what we're using this electrical energy to do, to open these channels. When these channels are open, all of the calcium comes flooding out. Okay? When we talk about the process of excitation, contraction, coupling, that's what this is. Okay? This is the link between the electrical energy of the action potential and the chemical reaction that leads to contraction of a muscle. Okay? So everything after this point is contraction. Now, why do we care about calcium? If we look a little bit closer here at our thin filament, right? Recall that we had sort of the primary body of this thing made up of a protein called actin, right? Well, as it turns out, there are other components to a thin filament. There's also a protein called troponin, okay? And troponin will bind to calcium, right? So this is the reason that we want to take all of the calcium out of the sarcoplasm when we're not contracting. Because if calcium is there, it will bind to this troponin. And that's an issue for us, right? Because what happens when we bind calcium and troponin together is that the other part of this, right, this little rope that we're attached to here, made of a protein called tropomyosin. At rest, that tropomyosin is covering the active sites on the actin proteins. They're covering the parts that will bind to other things, right? So as long as tropomyosin is where it's at rest, the thin filament can't interact with anything else. Okay? Troponin binds to calcium, it changes the shape of troponin, and it moves tropomyosin out of the way. And now we've activated, essentially, the actin of the thin filament, and now we can bind to other things. And that binding to other things is what's actually going to cause the contraction itself. Okay. And what we're going to do is we're going to take those thick filaments, right, those myosin heads that we talked about on those thick filaments that are reaching out towards each of the 6 thin filaments around it. What we're going to do here is we're going to enable those heads to bind to the thin filament. Okay. We are going to be able to make a connection between those two. And that connection is called a cross bridge, right? It's bridging the gap between the thick and thin filaments. And the process by which those cross bridges form, move the thin filaments, and are broken again is called the cross bridge cycle. Okay. So what we're going to do now is we're going to take our thin filament from before, okay? We're going to give it some more context. We're going to bring the thick filament in here also. So at rest, A normal condition here, the thin filament is set up, right? All of the active sites are blocked. There's no calcium in the place. Okay? The myosin head on the thick filament is held back, being pulled backward from its normal resting position, being pulled backward and held backward through the action of ATP. So when we talk about skeletal muscles, when we need ATP, right, we need ATP for contractions, okay? But we need them not to move the filaments, but to stop them from moving. We will need ATP for a skeletal muscle to rest, okay? Not to contract, but to rest. When we bind ATP to this head, okay, and we pull it back into what's called the cox position, okay? Typically, we create a molecule of ADP, diphosphate to the triphosphate. Phosphate from the original part is still here, but it's now moving to the spot where we can move it along. Calcium comes in, shifts. We allow the tropomyosin to move out of the way. We expose the active site, right, where we'd stopped on the previous slide. Now we're going to break this ATP off. It's going to go away. And as we allow it to go away, that myosin head is being pulled back and held basically under tension, springs upward. Right? It pops back upward. As it pops back upward, it binds to an active site on the thin film. Okay? Momentum from being held backward and allowed to spring forward is going to take it, grab it, and pull it. So we're going to continue to move. Moving the thin filament this way relatively quickly, pulling those Z discs closer together. We'll do this again and again and again and again and again and again and again. Okay? Thousands of times. Each one of those little pulls moving those Z discs a little bit closer together. Okay? Repeated across the entire filament, repeated across the myofibril, it brings the entire muscle fiber short. To bring us back to rest, we bind ATP to the myosin head, pull it back. We spend ATP to take the calcium that's free floating out there and to pull it back into the sarcoplasmic reticulum. If there's no calcium, there's no cross bridge. There's no cross bridge, there's no contraction. Okay. Reset for the next time around. And that's how contraction works. That's how sliding filaments slide. You might have heard, almost certainly have heard, of a condition called rigor mortis. Okay? This idea that a dead body is relatively stiff and immobile. Yes, but only for a brief window of time. Usually dead bodies are pretty floppy, surprisingly so. What rigor mortis is, right? Remember that we need ATP for a muscle to relax, right? To break cross bridges, we need to spend ATP. Okay? When you die, you're dead, okay? But the chemistry in your cells continues to happen. Chemistry doesn't care if you're living or dead. Chemistry just continues on, okay? And so the ATP in your cells will continue to get used, right? Eventually, though, you're going to run out of ATP. Okay? And this is going to happen within not a very long time, about 3 or 4 hours, give or take. Okay? So for the first couple hours post-mortem, you're full-on weekend at Bernie's flopping around. Okay? At about 3 hours till about maybe 12 hours, so again, a really brief window of time, okay? You're out of ATP, and so you can't break those cross bridges. So your muscles cannot move. They are stuck in the position that they're stuck in. Okay? That's what rigor mortis is. However, the other thing that people sometimes don't know about being dead is that you begin to rot essentially immediately, right? You are always kind of teetering on the brink of just rotting like a vegetable in the back of your fridge. As soon as you die, that process begins right away. Okay? And within about 12 hours, the components of your cells, muscle cells included, have decomposed to the point where those cross bridges are no longer even present. Okay? And so you go back to being floppy again. So one thing you can do if you're an ME, you can look at the condition of a body, and the pre and post rigor mortis conditions are substantially different. Let's put it that way. You can look at a body, you can manipulate a body, you can give a rough estimate for time of death if you're within kind of that period of time. Okay. So the neat thing about how muscles work is that essentially if you understand how one muscle fiber contracts, you understand how the whole muscle contracts. Okay. The basic principles of contraction here basically just scale up. Okay. And what we're doing here through a contraction is we're generating a specific kind of force called muscle tension. Okay? And muscle tension is the force that theoretically is able to perform work, right? To move a load a certain distance. That's what work is in a physics context. Okay? Whether or not it actually does that or not is dependent on the force and the load and all these things we'll talk about. But one muscle fiber generates tension, an entire muscle generates tension. They do things basically the same way. One of the confusing terms that we use in here is the term contraction. Okay. Typically, when we talk about a contraction, right, in sort of a colloquial, common-day sense, right, we're talking about something getting shorter, right? A contraction in a grammatical sense is when you take two words and you make them one word that's shorter than both of them combined. Here, in a physiological sense, a contraction does not necessarily imply any shortening at all. In fact, a contraction can imply a lengthening. Okay. Contraction is simply the term that we use to describe the physiological process that generates tension. Okay. So a contraction, probably like you're thinking about, right, in your head right now, you think about a muscle contraction, one where the muscle does get shorter, right? Or something moves. This is an isotonic contraction. Okay. And this happens when the tension, right, the force, is greater than the load that you're trying to move. So if I go over here to this card table and I pick it up, or I try to pick it up, I can pick the table up. It's not like it's an impressive feat or anything. I can pick it up, right? If I went out to the parking lot and I tried to pick up my car, that ain't going to happen. Okay. The difference between those two things happening, the difference between those two loads moving, is the amount of tension I can apply to it. Okay. The amount of tension I can generate. So if I can exceed the load, then I have an isotonic contraction. If I can't, if I go out to the parking lot and try to lift my car, right, it's not a big car, but I still can't pick it up. Right? Now we're talking about an isometric contraction. I'm generating force, right? I'm having a contraction, okay? But that force, that tension that I'm generating is not enough to move, in this case, the car, and so my limb doesn't move. My muscle doesn't shorten. Okay? It's still a contraction, but it's not moving. Okay? Then there's a special kind of isotonic contraction I'll talk about a little bit later on, where the load exceeds the force and the muscle lengthens. So those things can happen too. Okay, so the difference between these two things then is the amount of force being applied. Right? So one of the things that we have to think about when we talk about how muscles work is how do we vary the amount of force that we apply? How do we control the amount of tension that we generate? Right? Because there's a difference between going and picking up a kitten, right, and being very gentle with it, and going and picking up a brick. Right? You use different amounts of force for those two activities. Well, how do you generate those different amounts of force? With the same muscles. Right? One big part of how this works is how the wiring works in your muscle. Okay? Remember, you might have 1 million different muscle fibers in a single muscle. They ain't all connected to the same neuron. Okay? You're going to have many different neurons in the same muscle. One nerve, but many neurons. Okay? And each of those neurons is only going to go to a handful of the muscle fibers in it. So one neuron and all of the muscles that control, muscle fibers it controls, okay, called a motor unit. Okay? And so one of the things that's going to be different about different kinds of muscles, right, is how many motor units do you have, right? The muscles of your hands, for example, that move your fingers, okay, that's a case where we have a lot of motor units, relatively few muscle fibers per neuron. Okay? This is going to give us really good, fine control over contraction, okay? Because we're going to be able to use some, but not all, of the muscle fibers. So imagine you've got a muscle with 100 fibers, right? If you have 2 motor units, and each of those motor units controls 50 of those fibers, your only choices are 0% contraction, 50% contraction, or 100% contraction, right? Because you can only have one neuron or both neurons or neither neuron. If you have 10 neurons going to that same muscle, each of them with 10, right? Now you've got a lot more control because you can do 0 or 10 or 20 or 30 or 40 all the way up to 100. And so the number of motor units and the number of muscle fibers each motor unit controls plays a big role in your ability to control the amount of force that you apply with a given muscle. So here, for example, we've got one fascicle pulled out here, okay? 2 different neurons, one in purple, one in red, okay? So for example, motor unit 1 here, the purple neuron, this neuron, this muscle fiber, and this muscle fiber. Motor unit 2, right, is this red neuron, this muscle fiber, and that muscle fiber. So again, it's just a means of controlling part of a muscle without controlling the whole thing. Gives us a lot of variability in what we do with them. One other thing that's true about how motor units are put together is that typically one neuron is connected to muscle fibers across the entire muscle. Okay. This prevents us from having situations where like one side of a muscle contracts and the other one doesn't, right? If all of the motor unit is on the right side of your muscle and there's nothing on the left, you'll get this weird asymmetrical contraction. We don't want that. Okay. The other thing that we're going to be able to do with these motor units, it's very, very helpful for us, is when we have a sub-maximal contraction. Right? So we're not lifting something with the maximum amount of force we've got. Say we're only lifting something that requires 20% of our total potential. Okay. What we can do with these motor units is we can switch them on and off to meet that 20%, right? And give them ones that have been working a little bit of a rest. So again, we go back to our example of our muscle with 100 motor units in it, right? Or 100 muscle fibers and 10 motor units. For that 20% example, right, we turn on 1 and 2. And we hold those for a little while. And then we give 1 and 2 a break, we turn on 3 and 4. Then we give 3 and 4 a break, 5 and 6, and on down the line. Eventually we can circle back to 1 and 2 again, right? By enabling us to give muscle fibers a rest, right, by switching on and off motor units, we prolong the capacity to maintain a contraction. Right? So it gives us control over force, and it allows us to maintain contractions for longer, or submaximal contractions. Okay. When we look at the actual force applied by a muscle fiber or a muscle altogether, we're going to do so based upon something called a muscle twitch. Now, a muscle twitch is a laboratory-only kind of thing, okay? In that it is the response of a motor unit to a single action potential. Okay? In reality, you never get one action potential. You get 2 or 3 or 100. You never get just one. Okay? So that makes this kind of a lab-only situation. But we can use this to understand how real-life contractions actually happen. Okay? The diagram that we're going to get out of this is called a MIOL, measurement of force over time. So when we look at this myogram here on the next slide, we're going to be able to see three different phases, three different periods in this contraction, right, relative to when the action potential arrives. So the action potential is going to get there, and for a long time, nothing's going to happen. This is going to look like nothing's happening, right? We're not going to start generating any force for a little bit of time. It's called the latent. The reason we're not going to have this latent period, the reason we have this little gap, is all of the chemistry we just got done talking about, right? The action potential gets there, okay, it's got to travel down the sarcolemma, it's got to get to a T-tubule, it's got to open those calcium channels, calcium's got to bind to troponin, troponin's got to move tropomyosin, myosin head's got to bind to the thin filament, right? All that's just got to happen, right? It's fast, but it's not instantaneous. Okay, so we've got this little bit of a lag period built in there. Following that period of contraction, once we start forming cross bridges, now we're generating force. Okay, and so our myogram is going to go up. The curve of our myogram is going to go up. And that's going to be the period of contraction. We're actively building force. We're actively generating force. Okay? Once we peak out, once we hit our peak force generation for that particular twitch, we're now going to begin slowly relaxing that muscle out. Okay? Force doesn't immediately fall to zero, it gradually falls to zero. So we enter this period of relaxation. Okay? The key thing here with the muscle twitch is that this is typically pretty fast, this is typically pretty slow. The length of time spent in this phase is much greater than really these two combined. And that becomes very, very important for us physiologically. Okay? So let's look at a myogram here. These are milliseconds, so it's really, really not a long period of time. Really. Here at time zero, we have our action potential. We have this little phase right here, where again, we're doing all of the stuff leading up to cross-bridge formation, our latent. Once those cross bridges are forming, we generate force, we generate tension. You see it reflected here in our myogram. Reach maximal tension. Now we begin the slow decline back to zero. This period of relaxation. Not every muscle is going to have the same response, right? You might have heard the term fast twitch muscle or slow twitch muscle, right? Different muscles are going to have, everybody's got the same latent, right? That's built in, that's chemistry, that can't change. But the time it takes to reach maximal contraction and the time it takes to return to zero can vary quite a bit between different muscles. And the reason behind that is entirely down to what we're asking those muscles to do. Right? So if we look at three different muscles, so one of these muscles we've already seen, right? Our old friend, lateral rectus muscle behind. Okay? Your eyes have to make relatively rapid movements. Right? They need to be able to track objects moving across your field of vision. Right? Very, very fast response. Okay? So the entire period of contraction is really, really very short. Contraction period, the relaxation period, compressed, very, very small amount of time. These other two muscles you might not know right now, but we will learn later in class, right? These are muscles in the back of your calf. Okay? There's gastrocnemius, which is the big one in the back there, and there's soleus, which is sort of the flat one. Okay? Gastrocnemius and soleus, while they operate the same joint, have roughly the same origin and insertion. Okay? Do different things for it. Gastrocnemius is the muscle you use when you walk, when you run, when you jump. Okay? So for high intensity kinds of activity. Soleus is the muscle that maintains your posture. Okay? So gastrocnemius, because it has that high intensity, rapid kind of activity we want out of it, has a relatively fast twitch. Not as fast as the eye muscle, but it's still pretty fast. Soleus, because its only job is to keep you from falling over, right, doesn't have to have a fast twitch. And in fact, has a relatively long period of both contraction and relaxation. Again, owing to the different tasks that we ask each of these muscles to do. Not every muscle is built the same. Now, when we get into the realm of reality, okay, so outside of the laboratory universe, and we have multiple action potentials arriving for each contraction. Okay. What we're going to be able to do with those multiple action potentials arriving, okay, is vary the amount of force, vary the amount of tension being applied. These are called graded responses. So we talked about graded potentials in the nervous system, right, where we had kind of a weak and maybe a little stronger, a little stronger, a little stronger, not stronger, up to full, right, the whole range, right? Unlike an action potential, which is on or off, right, more of a dimmer switch kind of a situation, right? Graded muscle responses, same basic idea. Right? It's not on or off, it's anywhere from being totally off, being maximal contraction with every possible step in between. Okay? The gradation happens through two different means forms. Right? One of them we've kind of already talked a little bit about, strength and stimulation. One of them we haven't really talked about, and that's frequency of stimulation. So what we've seen so far, right, with our monograms that we've seen so far, the twitch that we've looked at before, right, this is one action potential, right, just a single one. And that one action potential generates one single event. Period of contraction and a period of relaxation. What happens if we start having more action potentials? What happens when we have a stimulus and then another stimulus and another stimulus? What we're going to do is we're going to essentially add those together. Okay? Process we're going to call wave summation, right, or temporal summation. And summation here just meaning, again, adding together. One of the things that we rely upon here for our understanding of how this works is that long relaxation period. Okay? When we have a twitch, when we have a single action potential, right, that single action potential generates a certain amount of force, a certain amount of tension. So say that tension applied is half a Newton. Okay? If we start at 0 and we have an action potential, we can generate half a Newton of force. What happens if we don't start at 0? That's where wave summation comes into play for us. We start at 0, we have our half a Newton, right? But we're slowly relaxing, we're still generating force. And now we have another action potential. We don't start at 0, that we start up here. And we add that half a Newton. Right? Then we relax a little bit more. And we add another half a Newton. So on and so forth. As long as we have these action potentials occurring within the relaxation period of the one prior to it, we can generate more force. We can add all of that force together, right? And get significantly more tension, right, than we would from a single action. Now what we're seeing here is an example we call unfused tetany or unfused tetanus. Now tetanus you might have heard of as something that you have to go get a shot for if you step on a rusty nail, right? The reason we call that tetanus is that particular bacterial infection can cause permanent kind of contraction of a muscle, okay, or tetanus, right? All that means, right, is the condition of contraction of the muscle. So here we've got it called unfused or incomplete tetanus. Because if we look at the wave of our myogram, right, there are peaks and valleys and peaks and valleys and peaks and valleys. Okay? It's not one solid, consistent curve all the way through. And with unfused tetany like this, right, this is good because this builds in relaxation periods, right? Builds in little rest periods. It's not something where we're going to work until failure. On the other hand, right, if we don't have those little rest periods, if the action potentials come so fast that we don't actually get to slowing down, right? The next action potential occurs here or here or here or here, anywhere where we're still climbing, there's going to be no relaxation, right? And so we're going to get instead what's called fused or complete tetanus. Right? And the thing with that, of course, is that without those relaxation periods, without those rest periods, we work until failure. Right? Muscle fatigue results from it. So when we talk about muscle fatigue, we're talking about something very, very specific. We are incapable of contraction. We have damaged the sarcoplasmic reticulum. We have completely thrown our ionic balance out of whack. Right? One or both of those conditions, we are chemically incapable of generating a contraction. Right? We have worked until we've reached that point. And fused tetanus looks something like this. Again, one complete solid curve, right? You work until failure relatively. The other way we can, of course, change the amount of force applied is through the change in stimulus strength. And we've talked a little bit about this as a concept already, right? This idea here of recruitment. Okay. Remember, we don't have to contract every muscle fiber every time we have a contraction. In fact, we usually don't. Right? But the more that we bring in, the more strength that we've got. Think about it like if you're out in the quad, you're having a tug of war, right? If you've got two people on your side, you're kind of up shit creek, right? But if you bring in your neighbors, you can't do this now, of course, because pandemic, but the idea, right? You bring in more people, the more hands on that rope, right? The more people you recruit for your team, the stronger you're going to be, right? The more motor units you bring in, the stronger that contraction is going to be. Okay. Within this notion of recruitment, again, we're going to talk about strength of stimulus here. Again, we're going to come up with this idea of a threshold. And our relationship to that threshold is going to determine what degree of recruitment we actually have. All right, so I'm just going to show you this picture because I think this picture makes the most sense in terms of trying to understand this. We've got three different components here, but they're all connected in columns. So we have stimulus voltage, right? Stimulus strength, basically. Each of these 10 columns here relates directly to the number of motor units being excited down here in the myogram that we see below. Okay. So as long as we don't have a sufficient stimulus to reach threshold, okay, as long as we're sub-threshold, so 1 and 2, nothing is going to happen. No motor units get excited, no contraction happens whatsoever. But once we reach threshold, like we do here at 3, now we have a relatively weak response, but we have a response. In this case, we have 2 motor units that are excited by this, and a relatively weak contraction. As we continue to increase the strength, we add more and more and more motor units until they're all excited. And As we add more and more and more and more motor units, the amount of force generated increases, increases, and increases. Now, of course, we're going to reach a point where everybody's already called, right? You've gone through your entire phone. There's nobody left to call, right? You have the most people you know pulling on that rope, okay? At that point, it doesn't matter how much stronger the stimulus gets, you can't have any more contraction because everybody's already on deck. Now, within recruitment, we have kind of a hierarchy of who we call, right? When you are moving, right, and you're calling your friends to help you move, who do you start with, right? You start with Biff, who's a linebacker, who has a truck, right? And you start and you get all the way down to the end to your 10-year-old cousin Willie, right? Can't pick up much, can be a help. Right? The way that recruitment works in muscles is the exact opposite, right? We're actually going to start with the smallest muscle fibers first. Because again, remember, when you are thinking about having a relatively weak contraction, right? You're picking up a kitten or a baby bird, right? You don't want to start off with Kung Fu grip. You want to ease into it. And so you're going to bring in the weakest ones first. If that's not enough, we'll bring in the medium side. It's only when you get to the very end of your limit, right? You're really pushing for generating force to try to move something. And you call it a big dog. Because again, you're not worried about having to gradually scale up at that point, right? You're already kind of up against it. You need as much as you possibly can get, so you're going to call in those biggest ones last. And this is called the size principle, or the size principle of recruitment. Right? Start small, work your way up to the big ones. All right. As far as types of contractions go, we mentioned some of this stuff a little bit earlier here, right? Notion of an isotonic contraction, right? The typical contraction you would expect when you think about a contraction, right? A muscle contracts, and in response, moves a load, the muscle shortens. Within this broader category of isotonic contractions, though, we have two different kinds. The one that you're probably thinking of in your head, right, which is a concentric contraction, where you generate a force, the muscle shortens, okay? But there is also an eccentric contraction. You're generating force, muscle's moving, but it's lengthening. When you think about concentric and eccentric contractions, think about doing a weightlifting exercise. Right? Think about doing, say, a bicep curl. Right? Start off the weight down here by your side, right? You bring it up, okay? This is the concentric part of this contraction, but you don't just let the weight drop. Good way to blow your elbow out, right? You want to waste some energy in the process, right? You want to slowly let that weight back down. You want to slowly relax that muscle. You're generating force, you're not just letting the weight drop, right? But you're also not shortening that muscle. That muscle is lengthening back out again. That's an eccentric contraction. For a typical concentric kind of contraction here, again, remember the important relationship that we've got is between force and load. So as long as the force exceeds the load, the muscle will shorten. If we look at this in terms of a myogram and a resting length, so the myogram we've seen before, right? We're talking about your generating force, right? Contraction period, relaxation period, and so on and so forth. The one at the bottom here might be a little confusing at first. This is resting length, right? So as we go down, as we drop from here down, that just means the muscle is shortened. we're going to have our contraction, right? We're going to begin generating force, but we're not going to do a damn thing until we actually cross the amount of resistance that we've got to cross. Through this entire part of this is an isometric. Muscle's not moving because we don't have enough force there. Only when we actually cross this barrier, when force exceeds the load, do we actually be short. Of course, then the opposite of this, right, is that isometric contraction, right? Generative force, not enough to move anything, so the muscle itself doesn't move. Right? So go back to the example we had before, right? Now we double the weight on the end of this muscle. Force generated here, not enough to move 6 kilograms, enough to move 3, but not 6. So the weight doesn't move, the muscle doesn't move. We're still generating force, right? Our myogram is still showing that we're having a contraction. But because the force that we're generating never crosses the line of resistance, never exceeds the amount of weight or other sorts of work required here, muscle never contracts. Right? We never change that resting length because we never exceed the load. All right? Last thing for today, notion of muscle tone here. All of the muscles in your body, all the skeletal muscles in your body anyway, are continuously partially contracted. Okay? You are cycling through motor units all the time. Okay? Not like in a really sort of massive sense, not in like a full-on contraction, but just a little jolt, little spark. Okay? And the whole point behind that, right, it's entirely reflexive. The whole point behind that, right, is to maintain the muscle itself. If you don't have neural connection to the muscle, if you don't have stimulus to the muscle will wither. Okay, but it's kind of like stretching before you go do something, right? Keeps you kind of in a state of readiness. Prevents you from being cold, so to speak, right? And having a muscle tear or a muscle sprain as a result of sudden action. Okay, all right. Have a good weekend. I will see you guys on Monday.